Hello, everybody. My name is Nasim Mohammadzadeh, and I'm a senior at Paul Laurence Dunbar in the Math Science and Technology Center. And today I'm going to be talking to you about the effect of insulin-like growth factor 1 on Reelin expression in the dentate gyrus after traumatic brain injury. Now, that's a really long title to unpack, but before I start, I'm going to be giving you guys a brief overview of my project. So first, I'm going to be giving you a little bit of background information along with my introduction to help you understand my project a little bit better. Then I'm going to be talking about my questions and hypotheses and the methods that I went to test my hypotheses, which were tissue selection, immunohistochemistry, and image and data analysis. Then I'll tell you guys about my results, my conclusions, talk about some future research that's possible, and then give some thanks with my acknowledgements, and then leave you with my sources. So let's start talking about my background information. So why does damage to the hippocampus cause issues? So the hippocampus is a portion of the brain that's most responsible for learning and memory. And traumatic brain injury, or TBI for short, results in neuronal damage and death in the area. Now, this loss of neurons has actually been attributed to a decrease of cognitive abilities in the brain, which is why we see some short-term memory loss after traumatic brain injury that impacts the hippocampus. And something also that's really important about the hippocampus is that it's one of two parts of the brain that's responsible for a process called neurogenesis. Now, what is neurogenesis, you might ask? Neurogenesis is the process of developing functional neurons from progenitor cells, and it occurs in the dentate gyrus of the hippocampus. And something important about neurogenesis is a process called neuronal migration. Now, when the neurons are still progenitor cells and they're immature, they lie in the subgranular zone of the hippocampus. And once they've matured into granule cells or functional neurons, they travel to the granule cell layer. And this is a process called neuronal migration. And in the past, neuronal migration has been proven to be responsible for converting short-term memories into long-term memories. So this process of maturation and migration helps convert short-term to long-term. And now we want to look a little bit further into what happens in neurogenesis with traumatic brain injury. In figure A, you can see a healthy process of neurogenesis. You can see that these progenitor cells that lie in the subgranular zone, or the yellow layer right here, are pretty common. They're the blue ones. And after they're done maturing, they travel into the granule cell layer and neuronal migration. And as you can see, they lie kind of on the lower layer of the granule cell layer. And that's in healthy neurogenesis. Now, after traumatic brain injury, we can see that neurogenesis is actually dramatically increased. You see a lot more of those blue dots in the subgranular zone, and you see that there's a lot more of them in the granule cell layer as well. But something important that happens after traumatic brain injury that is seen as problematic is the abnormal migration of these mature neuron cells in neuronal migration. So we can see that they're really relevant in the molecular layer and in the Hilar layer, and they also kind of tend to travel into this lower layer of the granule cell layer, or the outer edge of it. And this is really bad because this abnormal migration can be seen to cause post-traumatic epileptic symptoms, which is something that we want to prevent, which is why I decided to study this a little bit further. Now, what is insulin-like growth factor 1, or IGF-1 for short? IGF-1 is a hormone in the bloodstream, and it's a hormone that kind of regulates the production of hormones, so it's a hormone responsible for hormones. And in the past, its overexpression in mice brains has seen to increase the number of new hippocampal neurons and kind of restore a little bit a healthier neurogenesis in the area. And this has, as a result, caused to improve memory and learning ability of mice after traumatic brain injury. Now, I want to see if this insulin-like growth factor 1 had any effect on a protein in particular called Reelin. Now, Reelin is a glycoprotein that's present in the hippocampus, and it's a protein that isn't necessarily researched as much as we'd like it to be. In the past, we've seen that the lack of Reelin in mice that are naturally absent of it, or Reeler mice, has been, there's been a lot more abnormal migration in mice without Reelin. So we can see that there's a lot more epileptic symptoms in mice without Reelin. And as a result, it's been thought that Reelin is kind of a guiding factor for neuronal migration, but it's not necessarily proven 100%. There's been a couple of studies, but nothing that is actually relevant yet. And something that's not really known are the effects of traumatic brain injury and insulin-like growth factor 1 on Reelin expression, which is where my question arises from. So my questions are, what are the effects of traumatic brain injury and what are the effects of insulin-like growth factor 1 on Reelin expression in the hippocampus? And my hypotheses were that Reelin expression in the dentate gyrus would actually decrease after traumatic brain injury, and that insulin-like growth factor 1 would help restore the healthy Reelin expression in the area. And now, that being said, let's get into a little bit of how I tested my question. So a disclaimer, I wasn't actually able to handle any of the mice in hand because I'm underage. So I was just given their tissue after all of this process was completed. So there were 14 mice that received a controlled cortical impact and 6 mice that received a sham injury. Now, those 14 mice that received a controlled cortical impact were given an impact on the ipsilateral side of the brain only, which is the right side of the brain, and the contralateral side, which is the left side, was remain untouched, so we could compare the injured side to the uninjured side. There were 14 that received injury and 6 that received a sham injury, or in other words, just a craniotomy. And after traumatic brain injury and after the procedures were completed, the mice were either received an intranasal administration of insulin-like growth factor 1 or no administration of insulin-like growth factor 1 for 7 days. So we had 7 animals with injury and IGF, 7 animals with injury and no IGF, 3 animals with a sham injury and IGF, and 3 animals with a sham injury and no IGF. After they were administered their drugs, they were euthanized, and the mice brains were sliced by me, and the 3 epicenter pieces were selected. So I selected 1 from the most rostral section of the brain, 1 at the direct epicenter of the brain, and 1 from the most caudal section of the brain. And this is important because the hippocampus is visible in different amounts in those 3 sections. So after I selected my tissue, I then continued with the immunohistochemistry, and I wanted to stain for Reelin positive cells. And to do that, I used primary antibodies to bind to Reelin positive cells, and I used secondary antibodies to bind to the primary antibodies and amplify the expression of those Reelin positive cells. So once I administered my primary and secondary antibodies to the tissue, I then had to have a DAB staining solution to stain the Reelin cells so they were actually visible against the tissue background. After that, I uploaded all of my tissue images to a software called Halo, and Halo was my best friend during this process because it helped me automatically quantify Reelin positive cells in the areas that I wanted to study. And those areas in particular were the molecular layer, the granule cell layer, and the Hilar layer. So I annotated each of those layers, and then Halo automatically counted all of those Reelin positive cells based on the staining intensity of the image. So after it did all that for me, I actually had to analyze the data a little bit. So I received the number of Reelin positive cells, the average cell area, the total cell area, and the total tissue area of the Hilar layer, the granule cell layer, and the molecular layer. But a data that I actually focused on was the Reelin positive cell density. So that would be the real number of Reelin positive cells per millimeter squared, in particular for the Hilar layer, the granule cell layer, and the molecular layer. Now, next, I actually calculated the standard deviation and the mean for each of the groups. So the sham injured and vehicle administration, sham with IGF, CCI with vehicle, CCI with IGF. And after all that, I used a one-way ANOVA test to validate my results and see what was actually valid. So this right here is an image of a sham mouse brain, and this in particular, images B, C, and D are important because that shows the 3 different sections of the brain that I was studying. So I took the rostral section, which is image B. You can see that the hippocampus is visible, but not as much as it is in the epicenter, which is figure C. And then figure D is the part that has the descended hippocampus, which is in the most caudal section or the most further back section of the mouse brain. And this right here is an image of the sections of the tissue that I decided to study in particular. The yellow layer is the molecular layer. The blue layer is the granule cell layer where we see this neurogenesis happening. And then the orange layer is the Hilar layer. So I wanted to see if Reelin would be present in the molecular and the Hilar layer because that's where we see the most abnormal migration in the first place. Now let's talk about some numbers. So for the Hilar layer, we could see that the contralateral side, which as I said, was the uninjured side, we see that there's nothing really significant that happened because it was untouched, which is something really good. We wanted to see that there was no difference here. However, for the ipsilateral side, we saw that there was a dramatic decrease of Reelin positive cell density in the injured animals in comparison with the uninjured animals. So this blue bar is the sham animal with the vehicle administration, and the gray bar is the injured animal with the vehicle administration. You can see that there's a big drop of Reelin positive cell density, and same with the sham IGF and CCI IGF. But something important to note is that although there was a little bit of an increase in Reelin positive cell density after insulin-like growth factor 1 administration, it wasn't necessarily significant. And the same thing happened for the molecular layer, or the granule cell layer, excuse me. We can see that on the contralateral side, there wasn't necessarily anything different happening, which is good. But for the ipsilateral side, we saw a significant decrease with traumatic brain injury, but no significant difference with insulin-like growth factor 1 administration. But it's important to note that traumatic brain injury did cause a decrease. For the molecular layer, there wasn't necessarily any significant data that we came across, which is okay, and I can talk a little bit more about what we can gather from this data in my conclusions. So as a whole, we noticed that traumatic brain injury caused a decrease in the amount of Reelin positive cell density for the granule cell layer and the Hilar layer, but not for the molecular layer. So this kind of validates my hypothesis, but not 100%. My hypothesis was that it would cause a decrease in the dentate gyrus as a whole, but it didn't. It only caused a decrease in the Hilar layer and the granule cell layer. And insulin-like growth factor 1 didn't actually restore healthy Reelin expression in the dentate gyrus and either in all three of the sections. But that's okay, because we can do a little bit more further research to talk about what this means. So we can look and see if other parts of the brain express the Reelin protein, and if they do, what is insulin-like growth factor 1's effect there. And we can also see if there's any other migrational factors excluding Reelin that are causing this abnormal migration to happen. We can look and see if there's any other treatments that could possibly help restore healthy Reelin expression in the area. But before we do that, we need to verify if the Reelin loss in the area is actually permanent after traumatic brain injury, because if it's not permanent, there might not even be a need for treatment. There's a lot more work to be done with this research. With that being said, I'd like to give a little bit of acknowledgements and thanks. So first off, I'd like to thank my mentor, Dr. Carlson Saatman. The grad student that worked most closely with me was Anthony DeSana, and he was a huge help through all of this. Dr. Benoit Joseph helped me with a lot of lab training and technical stuff going on in the lab. And the Spinal Cord and Brain Injury Research Center, which is the facility that I conducted this research, was very gracious and very willing to take me on. So I appreciate them for that. I'd like to thank the Kentucky Young Researchers Program, which is where I received my funding for this project. I'd like to thank my family and friends for supporting me through all of this and pushing me through the hard parts. And I'd like to thank Karen Young for offering me such great guidance throughout all of this. And I'd like to thank the Math Science and Technology Center for even giving me the community and resources to actually be able to do this project. So thank you. And with that being said, here are my sources.